Assalamualaikum warahmatullahi wabarakatuh and good night, the judges and the committee. So we are from team Ubat teams, stands for Universitas Brawijaya Aflatoxin Terminator. We'll present about our project in our participation of IG Indonesian League. The title of our project is the development of maize aflatoxin resistance using CRISPR Cas9 technology. First of all, let's talk about our problems. When we first try to look our problems in East Java, we're finding that East Java becoming the province with the highest maize production in Indonesia. And when we try to look at the global scale, maize is also becoming a pivotal crop in global agriculture. Faces critical issues in Indonesia due to aflatoxin contamination exceeding the safe levels. The data estimated monthly ingestion of Indonesian people is 33.7 ppb aflatoxin, and this issue becoming a very severe to health aspect, and it can cause a liver cancer and clotting disorder or even death in some cases. From this concern, is Java posing a threat to both human health and also the livestock. Aflatoxin produced by Aspergillus flavus fungus manifests as the hazardous gray-green moss on corn kernels. In post-harvest, aflatoxin amplifies rapidly. Despite Indonesia being the world's seventh largest maize producer, it has failed to capitalize on this advantage. Shockingly, in 2023, Indonesia only exported 0.001% of its maize production, indicating a waste of potential. The main hindrance lies in global standards for maize intended for human consumption. Europe, for instance, accepts only 4 ppb of aflatoxin in food, whereas Indonesia's current standard is alarmingly higher, at not more than 20 ppb. This contrast becoming a significant challenge because Indonesia cannot enter in the massive market because current standard is far from meeting the demand. Right now, the farmer is already using some method to prevent this problem, but surely not effective. Farmers use ammonia spray that can causing a new human health problem because ammonia is also an active substance. Talking about the research that have been done to solve the problem, researchers used to make a hybrid maize, but later this hybrid found activating other types of fungus. So we need a solution to prevent this problem, solution that works on gene level. Our goal in this project is we are aiming to develop an aflatoxin resistant maize to solve this problem by manipulating a specific maize genes using CRISPR Cas9 technology to produce maize that is safe to consume and improve its quality.
So here's the concept of our method. Genetic engineering testing will be carried out in silico, which will focus on testing DNA characteristic, plasmid design, and factor analysis that affect on-target and off-target in gene of interest. Here's the method that will be done. Starting from the plasmid design using PG3GB411 with 2 promoter used, the Cas9 promoter itself using UB promoters, and the sgRNA promoter is U3. Then in the plasmid, there is an ampicillin-resistant gene as a selection parameter for the success of the plasmid. Then proceed to the gene of interest that will be targeted is glycine-rich RNA-binding protein 2. With NCBI search is GRMZM2G001A50. GOI was obtained from previous studies using microarrays and prediction motifs from ZM-GRBRP2, which affect hypersensitivity response by inducing PGI activation, which causes cell death. Next. We will be testing by the two software CRISPR and CRISPR-P, where the sgRNA candidates in the two software will be analyzed as phase 1 screening and will be compared in terms of off-target score and on-target score. To maximize prediction of target in the candidate, sgRNA will be compared with other tools. Cas9 finder and microhomology score as phase 2 screening. The best sgRNA comparison results will be taken three types of sgRNA with cutting sites and PEM-NGT, so that the function of the software is to improve the prediction of the best sgRNA candidates. The best sgRNA results will be analyzed by prediction from existing secondary structure of sgRNA from the Crispr software. The results obtained will have sgRNA from with a total 23 base pairs, of which 20 sequences are guide RNA and 3 base pairs are PAM-NGT. The components of sgRNA itself represent the artificial cRNA and tracer RNA. Phase 1 screening 1 obtained off-target and on-target as many as 5 sgRNA candidates are the best from every existing software. Then from the 5 sgRNA will be taken again as many as 3 types of sgRNA. The best of the two then in the secondary structure component and of the existing sgRNA has a cRNA length of 20 base pairs. Tracer RNA as much as 14 base pairs. The results show that in the screening phase 2, the best indicator in microhomology is an indicator of the height of deletion that appears in target DNA and the absence of a complete repairing process resulting in gene loss in the wild type. The results show that the variation in the secondary structure of sgRNA exists as a support for the efficiency of artificial guide RNA as a genome editing. The indicators of the secondary structure are in the existing repeat and anti-repeat loops, and there are 4 artificial loops that play an important role. Repeat and anti-repeat loop itself is loop 1, which acts as a precursor to cRNA by catalyzing by the enzyme RNase 3 as activation of the RNA. Then loop 1 acts as a form of activation from CAS9 sgRNA DNA as a form of binding at the DNA cutting stage, and the presence of stem loops 2 and 3 plays a role in the stability of sgRNA in Cas9 in vivo. The existence of 4 loops serves as a presentation of the stability of DNA cutting on CRISPR as genome editing. In addition, the result of CRISPR-P use the Young system, which says that the loop parameters are presented with no or no presence of loop 1, then no more than 12 TBP, 7 CBP, and 4 EPB. The results prove that the best secondary structure in guide RNA 58 8 and 21 due to the stability of sgRNA in the presence of absence of the stem loops 2 and 3 as application in vivo tests. The presence or absence of loop 1 does not affect sufficiently on cutting sgRNA. After identifying the top 3 sgRNA possibilities, the sgRNA is inserted into the plasmid. The plasmid used in this work is PG3GB411, which has been paired. After identifying the top 3 of sgRNA possibilities, the sgRNA is inserted into the plasmid. The plasmid used in this work is PG3GB411, which has been paired with morphogenic regulators, allowing this plasmid to be improved and capable of increasing the efficiency of transformation in maize. The plasmid was inserted into the chosen sgRNA candidate before being introduced into Agrobacterium tumefaciens. The insertion of CRISPR-Cas into Agrobacterium tumefaciens was reported to have an 18 success rate. So here's the modularity. Vector details. In vector that we use derived from P gene tree containing CON-L, DLPL, and ampicillin resistant genes for stability. The promoter choice is UT promoter with strong tracer RNA expression recognized by monocot plants, and UB promoter originating from nice enhanced Cas9 expressions in efficiency. And for the efficiency improvement, UB1 promoter significantly improves CRISPR-Cas9 system editing efficiency. In terminator usage, we use nose terminator from agrobacterium used to stop or terminate Cas9 expression.
So, because we're gonna make a genetic modified organism, we also considering the ethical issues about this project, mainly the risk assessment. So we can see at the right picture, the risk assessment framework that we use is divided by 5 points. First, we gather the information basically from international and national laws, and then we evaluate the risk. And the third step is we develop a risk control strategy. Next, we choose and select the implement risk control measures. And the last one is review the risk and risk control measures. So there are three key factors that we're talking about in the risk assessment. First, the biological agent characteristics, including the transmission and the pathogenicity, and also the laboratory procedure or personal competency, and also the biosecurity. The third one is the endemicity, contagiousness, and availability of preventive measures. So here are the basically the points of risk assessment that we found. First, the biosafety implementation in Indonesia. In Indonesia, the biosafety implementation is still fragmented regulation across the ministries, and also its need for detail in risk assessment spanning regulatory elements. In education and preparedness, the Research Indonesia is early biosafety education at the higher education level, and also involvement of the researchers, biosafety and specialists, and responsible of the parties. Also, the GMO Indonesia is governed by the Government Regulation Number 21 of 2005 and advised by researcher in importation, assessment, and also the utilization. GMO in Indonesia still miss the pros and cons in this and in their development. First, there is addressing food shortage through genetic engineering. Second one is the supportive reason, including the pest resistant and the nutrition. And the third one is the opposition, safety concerns, impact on non-GMO producer. The example of GMO in Indonesia that we can found is the first one is BT corn as a widely accepted solution to pest problems, and also the adherence to regulation for biosafety in GMO development. So, talking about the solving local problems, we divided the points within five. First one is the maize health challenges. Next one is the solution of Ubat, economic impact, global market positioning, and also the economic use. So from these 5 points, we conclude that our project is openly accepted by the farmers in Indonesia. So talking about the user customer discovery, we found that the market niche for Ubat is very open, highlighted a market niche for Ubat in addressing fungal issues during maize planting and storage, and also the consumer preferences is also the Ubat is meet the consumer preferences, and the research agency is emphasized through the urgent need to combat the fungal infections in Java's maize varieties. And also the challenges with seed companies is, despite the effort, some seed production companies have declined discussion with our project. The real implementation, first we sourcing the genetic materials, and then we doing an analysis, assembling and in silico testing, in vivo and in vitro validation, strain selection, and the last one is shipping of modified maize seeds. In integrated human practice, we meet some few of researcher. Here's Miss Azeri and Mr. Riza. In Miss Azeri, we consider the type of maize that will be targeted of our project based on the field that found in the data that we found in the field. And also we're opening our minds to determine the method that we can determine by considering the development of transgenic plants in Indonesia. And we also discussing about the discussing to get a network that we can contact to discuss further regarding transgenic maize products. With Mr. Riza, we doing discussion to determine the type of CRISPR-Cas9 that will be used, namely knockout and apply the appropriate method and suggestion to find the three best sgRNAs in our project, such as looking at the MN of target score, mismatch nucleotide, and macromolarity score. We also have a discussion with farmers in Kediri, East Java, and we're doing online and offline discussion, and we determine that the intervention method that will be used to prevent the aflatoxin in maize. We also meet with International Federation of Biosafety, or for short IFBA, to discuss about the designing emergency protocols and dealing with BSL-2 accidents involving the use of GMOs and considering the safety form by looking at the risk assessment. Our public engagement project is divided by two. So we doing an Ubat goes to open house. In here we giving a education with this new student in Brawijaya University, providing them with hands-on with simple DNA isolation project, and then with simple gel electrophoresis that we are opening the minds of the new student in Brawijaya University. We also doing already doing a synthetic biology in maize development in Indonesia webinar to aiming introduce IG Indonesia leagues and our project to students and workers in the field of synthetic biology, and also discussing the regarding maize development and quiz in Indonesia with the expert.
So the conclusion of our research is development of maize aflatoxin resistant with CRISPR-Cas9 technology that we have carried out produced of a design of sgRNA, the plasmid that will be used, and determine the gene of interest that will be knocked out using CRISPR with in silico studies. We propose genetically modified maize with CRISPR-Cas9 by using the candidate of sgRNA and plasmid that we found that to enhance the resistance of aflatoxin in maize instead of using ammoniac spray. We consider this state based on the effectiveness and the minimal side effect of these methods.
So maybe that's all for our presentation. But before we closing the presentation, we also wanted to thank you to our sponsor, Mighty Plus, Pertamina, Krakatau, and also Bukit Asam that are already supporting our project so far. So maybe that's all for our presentation about our project in participating in IG Indonesian League. So thank you.